Hello viewers, welcome to my channel. Today we will discuss about some important questions related to antibody. This is one of the important topic in immunology. For CSIR NET, ICMR NET, GATE, NEET exam, the questions related to this topic is very important. Some brief notes are also provided here which will be beneficial for you. So stay tuned in this video. The first question, the antibody that forms after infection, option 1 IgM, option 2 IgG, option 3 IgA and option 4 IgD. The correct answer is IgM. IgM is the first antibody that made by the fetus. It is also the first antibody to be made by the virgin B cells when it is stimulated by the antigen. Basically, when our immune system encounters antigen or a pathogen, the B cells get activated immediately, releasing antibody into the bloodstream. The antibodies or immunoglobulins are of 5 different isotypes, such as IgG, IgA, IgM, IgD, IgE. Among these, IgM is the first to be produced following pathogen administration. Next question. Most antibodies are produced in the option 1 heart and liver, option 2 brain and meninges, option 3 lymphoid and spleen, and option 4 thymus and appendix. The correct answer is lymphoid and spleen. Antibodies are produced in the plasma cells and it is produced in endoplasmic reticulum of the plasma cells. These plasma cells are differentiated form of B cells. When B cell activated, they differentiated into memory cells and plasma cells. B cells activation occur in the secondary lymphoid organ such as spleen and lymph nodes. So these antibodies are produced in the lymphoid and spleen. Next, the minimum number of polypeptide chain in an immunoglobulin is option 1 2, option 2 5, option 3 4 and option 4 is 6. And the correct option is 4. Immunoglobulin or antibodies are a Y-shaped glycoprotein. These are produced by plasma cells in response to an antigen. It is typically 4-chain structure, 2 identical light chain and 2 identical heavy chain, forming the L2H2 structure. Next, the antigen binding site present on antibody molecule is known as option 1 epitope, option 2 paratope, option 3 aggretope and the option 4 is none of these. And the correct option is paratope. That is the antigen binding site that present on antibody is known as paratope. Whereas the antibody binding site present on antigen is known as epitope or antigenic determinant site. Aggretope is the region of an antigenic peptide that binds to an MHC molecule. Next, how many bands are seen when immunoglobulin G analyzed on SDS PAGE under reducing condition? Option 1 2 bands, option 2 4 bands, option 3 6 bands and option 4 is 1 band. And the correct option is 2 bands. These 2 bands are 1 band for heavy chain and 1 band for light chain. Immunoglobulin are made of proteins. It has 2 identical heavy chain and 2 identical light chains that are joined by disulfide bonds. The reducing agent in SDS PAGE is 2-markaptoethanol. This reducing agent breaks the disulfide bond, thus separating the 2 identical heavy chain and 2 identical light chains. The molecular weight of each heavy chain is almost 50 kilodalton, whereas the molecular weight of light chain of each heavy chain is almost 25 kilodalton. As you know that in SDS PAGE, the protein molecules are separated based on their molecular weight. The molecular weight of each heavy chain is almost 50 kilodalton and the molecular weight of each light chain is almost 23 kilodalton. So they separate in 2 bands. As a result, it produced 2 bands. Next question. The major immunoglobulin present in human serum is option 1 IgA, option 2 IgD, option 3 IgM, Option 4 IgG. And the correct option is IgG. 75% of serum immunoglobulin is IgG. IgG is also the major immunoglobulin in extravascular spaces. Whereas the major immunoglobulin present in human secretion is immunoglobulin A, that is IgA. Next, the strength of interaction between a single antigen binding site of an antibody and an epitope of an antigen is known as option 1 affinity, option 2 avidity, option 3 both affinity and avidity, and option 4 none of these. And the correct option is affinity. The avidity is the total strength of interaction between a multimeric antigen and a multivalent antibody. It is influenced by both valency of the antibody and the valency of the antigen. Whereas affinity refers to the strength of binding of a single antigen determinant site and individual antibody combining site. Next, the reagenic type antibody is option 1 IgA, option 2 IgD, option 3 IgM, and option 4 is IgE. And the answer is IgE, that is immunoglobulin E. This reagent also called the homocytotropic antibody or Prausnitz-Kutzner antibody. It is a type of antibody found in the serum and skin of allergically hypersensitive person and in smaller amounts in the serum of normally sensitive persons. Most reagenic antibodies are immunoglobulin E fraction in the blood. Antibody having high valency is option 1 IgA, option 2 IgD, option 3 IgM, and option 4 IgG. The answer is IgM. That is, antibody having high valency is IgM. IgM normally exists as pentamer but can also exist as a monomer. In the pentameric form, all heavy chains are identical and all light chains are identical. Thus, the valency is theoretically 10. Because of its high valency, pentameric IgM is more efficient than other isotypes in binding antigens with many repeating epitopes such as viral particles and red blood cells. As a consequence of its structure, IgM is a good agglutinating antigen. Thus, IgM antibodies are very good in clumping microorganisms for eventual elimination from the body. To achieve the same level of agglutination, it takes 100 to 1000 times less IgM molecules than the IgG molecules. To neutralize the viral infectivity, less IgM is required than IgG. IgM is more efficient than IgG at activating the complement. All these are due to their pentameric structure. Antibody which cannot activate the complement, option 1 IgE, option 2 IgM, option 3 IgG, and the option 4 is none of this. And the correct option is IgE. IgM is more efficient than IgG at activating complement. That is, both IgM and IgG helps in activating the complement. Complement activation requires to Fc region in close proximity. IgM is pentameric. As a consequence of its pentameric structure, IgM fulfills this requirement and is a glued complement fixing an immunoglobulin. Thus, IgM antibodies are very efficient in leading to the lysis of microorganism. Next, basophils have receptors for antibodies. Option 1 IgA, option 2 IgD, option 3 IgM, option 4 IgG. And the correct option is IgE. Fc receptor present on the surface of basophils bind IgE, which is responsible for the allergic reaction and macroparasite defense. Basophils are non-phagocytic granulocytes that function by releasing pharmacologically active substances from their cytoplasmic granules. These substances play a major role in certain allergic response. Next, antibody present in colostrum is option 1 IgD, option 2 IgM, option 3 IgG, and option 4 is IgA. And the correct option is IgA. Serum IgA is monomer, but IgA found in secretion is dimer. When IgA exists as dimer, the J chain is associated with it. When IgA is found in secretion, it is also another protein associated with it called the secretory piece or T piece. Secretory immunoglobulin A is sometimes referred to as 11S immunoglobulin. Unlike the remainder of the immunoglobulin, which is made in the plasma cells, the secretory piece is made in the epithelial cells and is added to the IgA as it passes into the secretion. The secretory piece helps IgA to be transported across the mucosa and also protects it from degradation in the secretion. Antibody present in secretion is option 1 IgD, option 2 IgM, option 3 IgG, and the option 4 is IgA. And the correct option is IgA. IgA is the major class immunoglobulin in secretion such as TR, saliva, colostrum, mucus. Since it is found in secretion, secretory IgA is important in local or mucosal immunity. IgA is the second most common serum immunoglobulin after the IgG. Normally, IgA does not fix complement unless aggregated. Next, the immunoglobulin which found in asthma at elevated level is IgT, option 2 IgM, option 3 IgG, option 4 IgE. And the correct option is IgE. IgA is the least common serum immunoglobulin since it binds very tightly to the Fc receptors on basophils and mast cells even before interacting with the antigen. Involved in allergic reaction. That is how it involved in the allergic reaction. As a consequence of its binding to the basophil and mast cells, IgE is involved in allergic reaction. Binding of allergen to the IgE on the cells results in the release of various pharmacological active mediators that results in allergic symptom. Immunoglobulin implicated in atopy or anaphylaxis and anaphylaxis. Option 1 IgD, option 2 IgM, option 3 IgG, option 4 IgE. And the correct option is IgE. Both atopy and anaphylaxis are type 1 hypersensitivity reaction. That is, it is the immediate type of hypersensitivity reaction. Anaphylaxis is acute, fatal and systemic reaction, whereas the atopy is non-fatal, chronic and localized form of reaction. Next, which antibody is found in elevated level in parasitic disease? Option 1 IgD, option 2 IgM, option 3 IgE, option 4 IgA. And the correct answer is IgA. IgE plays role in parasitic helminth disease. Since serum IgE level rise in parasitic disease, measuring the IgE level is helpful in diagnosing the parasitic infection. Eosinophils have Fc receptor for IgE, and binding of eosinophils to Ig-coated helminths result in the killing of the parasite. Next, J chain is present in option 1 IgD IgA, option 2 IgM IgA, option 3 IgA IgG, and option 4 is none of these. The correct option is IgM and IgA. J chain is the protein component of antibodies IgM and IgA. It is 137 residue polypeptide encoded by IgJ gene. The J chain is required for IgM or IgB to be secreted into mucosa. It helps in polymerization. J chain also plays the role in the activation of complement. Initial hypothesis stated that J chain was required for polymerization. However, it was subsequently found that IgM is able to polymerize in the absence of J chain as a both pentamer and a hexamer. However, both these exist to lesser number in organism lacking J chain. In such cases, there are also fewer IgA dimer. Next, antibody present in secretion is option 1 IgT, option 2 IgM, option 3 IgG, and option 4 is IgA. And the correct option is IgA. IgA is the measured class of immunoglobulin in secretion such as TR, saliva, colostrum, mucus. Since it is found in secretion, secreted IgA is very important in local or mucosal immunity. It is the second most common serum immunoglobulin. It does not fix complement unless aggregated. Thank you. Please like, share and subscribe to this channel.